Hello and good morning to Prof and everyone. So we are from Group 7 and we're going to present about the Brussels sprouts. I am Emmanuel Lamadana Polkipong with matrix number 197234 and I will start the presentation now. So the contents of our presentations will be introduction, bioactive compounds, health benefits and mechanism and analysis method. What is Brussels sprouts? So Brussels sprouts is a form of cabbage belonging to the mustard family Brassicaceae, widely grown in Europe and North America for its edible buds called sprouts. The scientific name of the Brussels sprout is Brassica oleracea variety Gemifera. As its name suggests, Brussels sprouts originated in Brussels, the capital of Belgium. The botanical family to which Brussels sprouts belong is the Brassicaceae, also known as the mustard family or cabbage family. So yes, cabbage, broccoli, cauliflower and Brussels sprouts are from the same family. Brussels sprouts resemble mini cabbages with diameters of about 1 inch and they grow in bunches of 20 to 40 on the stem of the plant that grows as high as 3 feet tall. They contain a sulfur-containing phytochemical called glucosinolates, which is responsible for the distinctive odor and bitter flavor. Perfectly cooked Brussels sprouts have a crisp, dense texture and a slightly sweet, nutty and green taste. Moving on to the nutrition facts of the Brussels sprouts, here I have provided you the nutrient contents for two types of Brussels sprout. One is the raw and the cooked Brussels sprouts. So moving on, we are going to continue with the bioactive compounds in the Brussels sprouts. So the main component that we're going to discuss about is the glucosinolates. It is a large group of sulfur-containing glucosides found in Brussels sprouts. So when the plant tissue is damaged, the glucosinolates are broken down by the enzyme myrosinase, which will release glucose and a complex variety of biologically active products. And the most important and extensively studied of these compounds are the isothiocyanates. It has been reported to have anti-cancer properties in human colon, prostate, and breast cancers. Next, these are the products of the glucosinolates. So the first one is the sulforapane. It is the isothiocyanate that is synthesized from the glucosinolate glucoraphanin, which has a lot of potential health benefits that will be discussed later. And the next one is the indole 3-carbinol. It is a breakdown product of the glucosinolates glucobrasicin, which displays valuable cancer preventive properties. And the next one is the DIM. It is the breakdown product of indole 3-carbinol, which can potentially prevent carcinogenesis and tumor development. And these are the minor bioactive compounds in Brussels sprouts that is also important, but we will not discuss it further today. So I'm going to just briefly introduce the notable bioactive compounds in Brussels sprouts. So in Brussels sprouts, there is vitamin K, and when you eat a half cup of cooked Brussels sprouts, you can already obtain 137% of the recommended daily intake. Vitamin K is essential for blood clotting and bone metabolism. Next is the vitamin C. Again, when you eat half cup of cooked Brussels sprouts, you already obtained 81% of the recommended daily intake. And vitamin C will act as an antioxidant that helps to promote iron absorption and immune function. Next is folate. It supplies 21% of the recommended daily intake and promotes proper fetal growth and development to reduce the risk of birth defects such as spina bifida. And as Brussels sprouts are green, of course it will contain chlorophylls, that is the green pigments, acts as antioxidants and has anti-mutagenic properties. Next is the alpha-lipoic acid, ALA. It supplies 12% of the recommended daily intake for women and 8.5% for men. ALA usually acts as the antioxidant again, which has potential effects on blood sugar and insulins and may improve or overcome diabetes. And the last one is the camphorol. It is an antioxidant that may reduce cancer growth, decrease inflammation, and promote heart health.
Hello everyone, my name is Yvonne Lee Wen, matrix number 19841. So I would like to present about the health benefits and the proposed mechanism of bioactive compounds present in the Brussels sprouts. So what are the health benefits of consuming Brussels sprouts and how does its bioactive compound will benefit our health? Brussels sprouts do provide a lot of health benefits to humans. First, it helps to protect against cancer with antioxidants and phytochemicals. For example, sulforaphane and isothiocyanates are the major chemopreventive metabolites. Besides, it helps to fight inflammation and heart disease. It also provides bone-building vitamin K, whereby vitamin K is responsible for keeping the skeletal structure healthy and help to prevent conditions related to loss in bone's mineral density like osteoporosis or bone fractures. Brussels sprouts supply a large amount of immune-boosting vitamin C to boost the immune system. Vitamin C acts as a protective antioxidant in the body, which reduces inflammation and cell damage. In addition, consuming of Brussels sprouts helps to restore digestive health. Just 1 cup of cooked Brussels sprouts will provide 4 grams of dietary fiber, which is important for maintaining digestive health. It also helps to protect eye and skin health as well as improve brain health, as the Brussels sprouts contain important powerful antioxidant vitamin C and vitamin A. However, we will only focus on the 2 major proposed mechanisms of bioactive compound found in Brussels sprouts, which are the prevention of cancer and the reduction of risk of coronary heart disease. So for the prevention of cancer, these are the complex mechanism pathway that show how the bioactive compound present in the Brussels sprouts, especially the glucosinolates, will protect us from various cancers. Glucosinolates of plant are classified as sulfur-containing, branch aliphatic, aromatic aryl, and aromatic indose. In nature, glucosinolates present in the cruciferous vegetable are inactive. Both the enzyme and the substrates are physically coexist in the plant but are separated from each other. However, the enzyme myrosinase is released along with the glucosinolate upon injury, for instance when we chew the vegetables. The enzyme will hydrolyze glucosinolates into isothiocyanates. In addition to this, human gut flora will also convert glucosinolates to isothiocyanates, which are available in serum for biological activity. It has been demonstrated that the chemopreventive potential of cruciferous vegetable is likely due to the glucosinolates and their secondary metabolites, for instance, the isothiocyanates. Isothiocyanates are able to activate phase 2 detoxification enzyme as well as to disrupt tubulin polymerization, inducing cell cycle arrest and the activation of apoptosis in cancer cells. Sulforaphane and its derivatives have demonstrated significant induction of phase 2 enzyme, including the glutathione S transferase and quinone reductase activity, which help in the metabolism of xenobiotics to prevent potent carcinogenesis. This enzyme also help in the acceleration and disposal of xenobiotic metabolites and the inhibition of phase 1 enzyme, which otherwise known to activate carcinogens. Several studies have also shown that sulforaphane induces apoptosis and inhibit the progression and metastasis of many cancers. Isothiocyanates are known to inhibit both androgen-dependent and independent prostate cancer cells through arrest of cells in G2M phase and induction of apoptosis. The major target of isothiocyanates in inhibiting cancer cells include induction of caspase, dependent and independent apoptosis, activation of c-Jun N-terminal kinase, and cell cycle arrest. Besides the induction and proliferation inhibition pathway, both isothiocyanates and sulforaphanes are known to inhibit the histone deacetylase activity in a number of cancer cells. Acetylation of histone is known to affect gene expression and is associated with the development of cancer. Compounds capable of inhibiting the enzyme activity, in this case the isothiocyanates and sulforaphane found in the Brussels sprouts, are known to arrest cell growth, induce cell death, or apoptosis, and inhibit tumor through alteration in gene transcription level. Studies have also shown that a woman's risk of breast cancer is related to the estrogen and progesterone made by her ovary. Being exposed for a long time or to high level of these hormones has been linked to an increased risk of breast cancer. Indole and diindole are the major bioactive molecules of cruciferous plant, including the Brussels sprouts, known to act on enzyme responsible for estrogen metabolism. Among the dietary indoles, 3,3-diindolemethane (DIM) is the most potent inducer of estrogen 2-hydroxylase activity. The net estrone level is reduced due to the estrogen blocking activity of 2-hydroxylated product, which is associated with lower risk of breast cancer. The cancer preventive potency of DIM may be due to its ability to maintain the higher level of 2-hydroxyestrone level. This table shows the active metabolites from glucosinolates, including sulforaphane, ireucine, and indole 3-carbinol, and their roles in preventing many types of cancer. For example, sulforaphane helps to prevent prostate cancer by inhibiting tumor growth and inhibit the cell cycle. Another glucosinolates, the indole 3-carbinol that present the Brussels sprouts, for instance, may prevent colon cancer, breast cancer, and prostate cancer through apoptosis induction and many other mechanisms. The second mechanism is on how the bioactive compounds present in the Brussels sprouts helps in reducing the risk of coronary heart disease. Oxidative stress plays a major role in the pathophysiology of cardiac disorder. High-level oxidative stress resulting from increased cardiac generation of reactive oxygen species, ROS, are thought to contribute to contractile and endothelial dysfunction, myocytes apoptosis and necrosis, and extracellular matrix remodeling in the heart. Epidemiological studies have indicated that cruciferous vegetable intake correlated with a decreased risk of coronary heart disease. Brussels sprouts contain isothiocyanates, which can produce a redox-regulated cardioprotective protein, thioredoxin. It was reasoned that its consumption could be beneficial to the heart. Actually, this is same to the previous slide on how the isothiocyanates are produced from the glucosinolates when we consume the cruciferous vegetables. However, this figure emphasizes on the production of the specific active metabolite, which is the sulforaphane that is responsible in reducing the risk of coronary heart disease. So, sulforaphane is the hydrolysis product of glucoraphanin, the main glucosinolates in Brussels sprouts. Under neutral condition, the glucoraphanin is hydrolyzed by the enzyme myrosinase to yield glucose, sulfate, and sulforaphane. So, for the mechanism on how the sulforaphane in reducing the risk of coronary heart disease is due to its ability of upregulating the expression of a range of antioxidant enzymes, including the glutathione S transferase that was correlated with the reduction in oxidative stress, which is the major culprit in cardiovascular complication. Glutathione S transferase is a major phase 2 detoxification enzyme that catalyzes the conjugation of glutathione with electrophilic compound, making them more soluble and facilitating their removal from the body. Sulforaphane can also induce glutathione S transferase cleavage of reactive oxygen species. Sulforaphane is known to protect the heart through initiation of survival signal and activation of redox signaling of thioredoxin enzyme, which are known to facilitate other protein by cysteine thiodisulfide exchange. Therefore, sulforaphane has been considered to act as an indirect antioxidant and a highly potent inducer of phase 2 cytoprotective enzyme. The resultant detoxification of electrophiles and oxidants can protect against carcinogens, oxidative stress, and inflammation.
I am Genga Tarani Gengan and my metric number is 196350. I would like to explain method of analysis. As we mentioned earlier, Brussels sprout contain various of bioactive compounds. So each bioactive compound has own method to analysis. So the first one, determination of vitamin C. determination of antioxidant activity, determination of chlorophyll and carotenoid contents, determination of total polyphenols and flavonoid contents, Proximate analysis, and determination of glucosinolates by high-performance liquid chromatography. Since we are focusing more about glucosinolates, let's see how the amount or quantity of intake glucosinolates in Brussels sprout were determined by the researchers. So here we found an example of research paper that already found the quantitative amount of glucosinolates by using high performance liquid chromatography, electrospray, ionization, tandem mass spectrometry. So let's see the findings of this research paper. Many methods have been proposed to analyze glucosinolates, a class of phytochemicals whose broken-down products are thought to be responsible for an improvement in health. However, few are quantitative and many are time-consuming. A selective and sensitive quantitative method for direct determination of intake glucosinolates was developed using liquid chromatography coupled to electrospray ionization tandem mass spectrometry with selected reaction monitoring detection. The sample, freeze-dried, were milled into fine powders using a mortar and pestle and stored at 20 degrees Celsius until ready for use. In brief, 40 mg of sample was added to internal standard, which is glucotropylene extracted in 1.5 ml of polypropylene capped microcentrifuge tubes, 70% aqueous methanol, and sonicated for 30 minutes at 70 degrees Celsius. After being cooled in an ice bath, the extracts were centrifuged. After that, negative ion tandem mass spectrometry was conducted to detect glucosinolates with selected reaction monitoring. The mass spectrometer was tuned by direct infusion of standard signaling producing maximum abundant precursor ion and fragment ion. A dual time of 0.08 seconds was used for each transition. Standard calibration curves were established by triplicate analysis, which is 7 concentration levels, and deviations from the mean calculated concentration over 3 runs were within 15% from the nominal concentration. At the lower limit of qualification level, the deviation was within 20%. For glucosinolates in vegetables, glucoberrin, signaling, progoiterine, and glucoerosine were quantified using their corresponding standard calibration curves because standards were not available for the remaining compound. Figure 1 shows the calibration curve for glucosinolate standard, where Table 1 shows that individual and total glycosinolate content in various of vegetables. According to the Table 1, Brussels sprouts contain more glycosinolates when compared to other cruciferous vegetables. So, the advantage of the proposed method includes analysis of individual interglucosinols rather than the conversion of desulfoglucosinolates and increased selectivity through the use of mass spectrometry and a tenfold improvement in detection sensitivity over conventionally used high-performance liquid chromatography techniques. Next, in research terminology, the population can be explained as a comprehensive group of individual, institution, object, and so forth with a common characteristic that are interest of a researcher. The health benefit of bioactive compounds in Brussels sprout were examined by a group of individuals for a research purpose. A British Journal of Nutrition, which from the Nutrition Society used study population method to identify the cross-sectional association between cruciferous vegetable intake, which is Brussels sprouts, and extensive calcification in the abdominal aorta. The research team used data from a 684 older Western Australian women. The study found that those with a diet comprising more cruciferous vegetables such as Brussels sprout had a lower chance of having an extensive buildup of calcium on their aorta, a key marker for a structural blood vessel disease. As we know, heart attack or stroke usually happens due to the narrowing of arteries, which is the result of what may be described as blood vessel disease. It is a condition that impacts our blood vessels, arteries and veins and can reduce the flow of blood circulating around the body. The reduction in blood flow can be due to the build-up of fatty calcium deposits on the inner walls of our blood vessels such as aorta. This research has proven that eating cruciferous vegetables such as Brussels sprout can reduce the risk of heart-related disease. This is because one particular thing found abundantly in these vegetables is vitamin K, which may be involved in inhibiting the calcification process that occurs in our blood vessels. As per the research, women who consumed more than 45 grams of Brussels sprouts every day were 46% less likely to have an extensive buildup of calcium on their aorta in comparison to those consuming little to no Brussels sprouts every day. In conclusion, Brussels sprouts contain a lot of health benefits. By eating adequate amount of Brussels sprout helps to maintain a healthy lifestyle. This is because they are high in phytochemicals, which make a major contribution to healthy sight and may reduce our risk of cancer and heart-related disease. So start from today. Please add adequate amount of Brussels sprout in your diet. The next slide is about the references that we used in this presentation slides. That's all from us. Thank you.